our weekly seminar, and the speaker today is Johannes Jaeger from various places. So complexity hubs, and yeah, it seems like Vienna is figuring heavily in on your creations, but you've also been in Barcelona for quite some time. So Yogi is developmental biologist, developmental geneticist, which is now trying to make the leap into philosophy, and that there should still be some trace of real science, so something there. It's not fully metamorphosed yet, or anyway, okay, so Yogi is...
Okay, thank you, Tom. That was a very accurate description. Let me borrow the stick from you. So this is going to be a philosophical talk. I don't know. I would call it theoretical biology based on empirical research. I hope it connects to the topic of the project because it is about modularity and their role in evolvability, I think. And it's basically just to give you, in case you get lost on the way, the basic point is that I think we need to think about how to detect modularity and describe it in networks in different ways than we usually do, especially in the field of evo-devo, but also in systems biology. Okay, so I'm going to talk about different ways of identifying and describing modules and how that connects to the role of modularity in evolvability. The empirical research that this is based on was done when I had a lab still at the CRG in Barcelona, and the theoretical work was mostly done with a PhD student, Berthe, who joined me at the Wissenschaftskolleg Institute for Advanced Study in Berlin and at the KLI, and also in collaboration with a mathematician, Nick Monk, a very typical picture of him here. From the University of Sheffield, and he's been doing this sort of work for a long time already. And the whole thing goes back to this sort of paper that I from Herbert Simon in 1962. It's a beautiful paper on because near decomposability of complex systems, very similar to what we call modularity today. I had a very interesting chat with Thomas this morning, and I know there's previous work on modularity, but this is sort of a defining moment. He contrasts two watchmakers that build these very complicated watches, and one of them succeeds and the other one doesn't, because one of them builds the watch mechanism in modules and the other one is one step after the other. He shows how you fail to assemble a complex system. So therefore, he says, basically, this sort of modularity is a universal characteristic of adaptive complex systems. And he has this beautiful quote. He says, everything is connected, but some things are more connected than others. The world is a large matrix of interactions in which most of the entries are very close to zero, and in which by ordering those entries according to their orders of magnitude, a distinct hierarchic structure can be discerned. So everything connects to everything, but some things are more important than others. And that sort of leads him to the conclusion that this sort of decomposability, that most of the interactions are zero, this is a requirement for complex systems to evolve in the first place. So he says this is not something that evolves itself, it's a prerequisite for any complex system to evolve. Okay, and now I have to revise my talk when you send me those references from the 50s. There's even earlier work on this. But of course, modularity is seen as a very, very fundamental sort of ingredient that you need for evolution. And luckily, we can see at the phenomenological level that most phenotypes are modular, have a certain independence from each other. This is a picture I've stolen from Günther's paper in 2007. But we could also take, for example, Lewontin's work in the 70s, where he says the characters can be treated as quasi-independent. So nothing is 100% black and white, but there's a certain independence of different character traits. So far, so good. I'm not telling you anything new, obviously. But there's this argument that character trait modularity is a prerequisite for evolvability. by limiting pleiotropic effects. Also nothing new here. You've all talked to Mihaela before. And so the question is, how does character trait modularity come about? Okay, so what are the causes of this modularity? So if you want evolvability, you need modular traits, so you need to grow them, and that is of course something that happens somewhere between the genotype and the phenotype, so we can take the metaphor of the genotype-phenotype space, and I'm going to interpret it here. I don't think there's many quantitative geneticists here today, but I'm going to interpret it not in a correlational way, so not as a correlational map, but as a map representing the processes of development physiology and metabolism that really sort of causally translate the influence of the genes into phenotypes. And we're going to be focusing primarily on what Waddington calls the epigenotype. And the epigenotype consists of the generative processes that take you from genotype to phenotype. Okay, so it's a very sort of, the dangerous word, mechanistic interpretation of the map in sort of terms of causal processes. Show you the causal influence of the genes on different or other epigenetic factors on the phenotype. So that's sort of the groundwork so far. So you can already derive a few things from this very sort of simple caricature that I've given you so far. One of them is the sort of fact that evolution must work on embryological processes in order to be able to change adult phenotypes. And this has been variously attributed to different classical embryologists, such as Walter Garstang, Gavin de Beer, Goldschmidt, and Waddington. It's extensively described in Ron Amundsen's book about the changing role of the embryo in evolutionary thought. I'm giving you a quote from Horder, and it says, in order to achieve a modification in adult form, evolution must modify the embryological processes responsible for that form. Therefore, an understanding of evolution requires an understanding of development. So, an understanding of development is fundamental to understand the structure of the genotype-phenotype map, and therefore it's absolutely fundamental to understand evolution. So, Amundsen calls this the causal completeness principle. So if you don't understand development, there is something missing in causal explanations of evolution. And that was also the topic of the summer school that we had about 3 weeks ago in Venice about mechanism. What is a mechanistic explanation in evo vivo? I prepared this for the quantitative geneticists, but very few here, so looking at you. So far, so good. So this is sort of my argument why I am doing evolutionary biology. Okay? But basically, what we're looking at is how the structure of the genotype-phenotype map in this sort of mechanistic interpretation influences the potential of the system to evolve, the disposition, and there's a group here that looks at evolvability as this sort of dispositional property of an organism, or a developmental, a sort of an abstract developmental system like a vertebrate limb. Yeah. Okay. So far, so good. So basically, you can conclude from the argument so far that if you have modular traits, and then you have to have a modular epigenotype. So there has to be some sort of modularity in the processes that generate those traits, and therefore you must have a sort of a modular structure in some way of the genotype-phenotype map. So to phrase it a bit differently, since phenotypic variability reflects variability in underlying generative processes, the genotype-phenotype map must have a modular architecture as well. But we want to pin down what we mean by modular, right? And I think we have a bit of a wrong idea about modularity at the moment in the field. So, of evo vivo, evo vivo people have this wrong idea, especially. Okay, this sort of argument has been made long before I'm making it here. There's a really nice paper by Joseph Needham 1933, where he talks about the dissociability of generative processes. He talks about physiology and metabolism and development and embryology at that point, which is another sort of early publication on sort of the modularity of processes in development, in the life cycle of an organism. So I want to start at a really basic level and just think about very briefly what modules actually are. Try to define modules. Lenny Moss writes, a module is a unit that has is a component part of a larger system. That's not controversial, and yet possessed of its own structural and/or functional identity. So this we often forget, that it has to have some internal coherence to be modular. It's not just any part of a larger system, but a part that can be distinguished from the rest of the system because it has a larger internal coherence than connections to other parts of the system. A module can be a biological entity, a structure, a process, a pathway, or a collective of entities with more internal than external cohesion. Okay? Modules can be delineated from their surroundings, and their behavior emerges as a result of the integrated activity of their parts. So not only does it have to be separable from the rest of the system, but it has to have some coherent activity. And I think that's a central point. That's very important. This is a point that's often forgotten. Modules must be extended in time. Okay, if they don't persist, then they don't have any effect. This seems trivial, but we'll come back to that later. I don't think it's a trivial point. And of course, to quote Günther and Lee Altenberg, modularity is always a matter of degree. Okay, it's not like organisms are made like Lego, out of Lego bricks, like the town hall of Oslo, but they are only to a certain extent modular. Quasi-independence is Lundin's term. Near decomposability is Simon's term, so it's always a matter of degree. It's not a clean black and white effect. And of course, please interrupt if you have questions or comments. So the traditional way to identify modules is by their function. You do genetics, basically. There's a famous review by Lee Hartwell and others in Nature in 1999. It doesn't say much, but it gives us a sort of an idea of what a module is. A discrete entity whose function is separable from those of other modules. And that's perfectly accurate and also perfectly useless in terms of finding modules. It doesn't really tell you how to identify modules at all, right? Okay. Whose function is separable from those of other modules. So traditionally what we do, of course, is we, for example, we do a mutagenesis screen and we try to identify genes that are implied in a certain function. My favorite example of that, of course, is the segmentation screen by Mr. Paulhart and Rieschaus, where they just saturated the whole genome of Drosophila and picked out all the mutants that affected the segmentation process. And you can see a little summary of that here. So here, my personal rendition of a Drosophila embryo. And not only could you identify genes that were involved in the segmentation process, but you could group them according to the phenotypes they were producing, which also turned out to be reflected in different expression patterns. So you have these maternal coordinate genes expressing proteins that diffuse through the entire embryo. And then you have, in the first step, the gap genes, large overlapping territories of gene expression that refine into the 7-stripe pattern by the parallel genes. And then in a 14-stripe pre-pattern that prefigures the morphological segments of the animal that form later in development. So basically you have a cascade of regulation where members, genes that are in the upper tier regulate genes that are in lower tiers, but not the other way around. It's very hierarchical, but at the same time, you have a lot of cross-regulatory interactions within the tiers. And so here's where the problem begins. We're trying, so now we've identified most of the genes that are involved in segmentation. If there's redundancy in the system, we have a problem. We will miss a lot of the phenotypes, the mutations, knockouts in mice, for example, don't cause any phenotypes because there's redundancy, right? There's some technical problems with that. But that's the general idea. But even if we manage to identify all these genes, so when we try to sort of classify them into discrete groups, and look in more detail at their interactions, it becomes very complicated. So this approach of identifying the function of genes purely through genetics is intrinsically limited. I'll give you one example for that. That again comes out of our own work. So here's an embryo that expresses the gap gene Kruppel, KR in the middle. So here's the head, here's the tail, and the Kruppel protein is expressed in the middle. And Kruppel is regulated by another gap gene called hunchback, which is expressed in the anterior of the embryo here. So if you mutate hunchback, what happens is that Kruppel expands to the anterior, where hunchback used to be. So how would you interpret that as a geneticist? Hunchback is a repressor of Kruppel, right? And if you do the opposite experiment, you overexpress hunchback. In a heat shock experiment, Kruppel is pushed further back to the posterior of the embryo. So you say, okay, you have more hunchback here, less hunchback here. It's a repressor. But at the same time, Kruppel gets very weak in this. So hunchback could also be at the same time an activator. And also, if you overexpress hunchback, the Kruppel domain expands greatly to the posterior of the embryo. So there's more Kruppel. So we cannot decide whether hunchback is an activator or a repressor, or maybe it's both. Okay, and so as soon as you have more than 2 or 3 factors, with these sort of interventionist approaches, you always have this sort of problem. So it's really, I mean, that whole point, I'm not just trying to confuse you and say doing genetics in flies is really difficult. I'm trying to tell you that there is an intrinsic ambiguity in such sort of approaches that are perturbing the system to find out. You're doing something funny, right? You're looking at mutants, but you're not interested in the mutants. You're interested in reconstructing the wild type behavior of the intact system. And the problem here is, of course, I said there's more factors involved. There's another repressor, KNERPS. KNERPS is very strongly repressed by hunchback. So if you take hunchback away, KNERPS invades the territory of Kruppel, and that explains why it's not expressed anymore. And if you overexpress hunchback, KNERPS completely disappears, which explains why it expands. So basically, whether this is a direct activation or an inhibition of an inhibitor is intrinsically undecidable in this case. Okay, and that's a fundamental limitation. Furthermore, if you look at a different sort of type of function. So Günther has suggested this sort of distinction between character identity and character state. If you think about this, these are just different functional roles that genes play in conveying attributes to a character. Okay, so the distinction is the following. Here you have insects with 4 wings. But in the Diptera, so in the Lepidoptera, you can beautifully see the forewings and the hindwings. They're both used for flying. In Diptera, the forewing is the flying wing, the hindwing is a haltear, which is a balance organ, and in Coleoptera, the hindwing is the flying wing, and the forewing is the elytra, which is used as a protective cover when the animal is not flying. So basically, the character trait flying wing is not about character identity, but about character state. So character identity is a broad sense homology based on continuity of lineages, forewings versus hindwings. So you can say the homologous character here is the forewing, which has different physical structures. So the physical structure is reflected in the character state. The characters vary in size, shape, and color. You know, wings, halteres, and elytra, but the specific state doesn't tell you anything about the identity or the homology of different characters. Okay, now this is the basis of Günther's theory, genetic theory of homology, because you can now go and try to identify the genes that either contribute to identity or state, right? And you do it in the same way again. You mutate the genes and you see what is affected, the identity of the organ or just the state of the organ. So this is sort of the traditional, very successful way in which you can do developmental biology in an evolutionary context in terms of doing genetics. Okay, so based on this, the idea then, of course, is that you can identify networks that are responsible for the identity of the character, not of the state, which are conveyed by a different set of genes. Okay, so this is very powerful, and this is what we want to do, right? So we want to find out which factors in a bigger network contribute to which function. And the problem is, as I said, that the approach is limited. So character identity networks are examples of functional models, yes?
But how do you not connect the function and the character?
Well, that's the problem, right?
I mean, in your work, you decide that you look at some trait, but that's not necessarily what would be considered a character, right?
No, there's lots of problems here, right? I mean, the first one, of course, Günther uses the word chin because Gould and Lewontin use the chin as an example of something that's not a character trait. The human chin is made out of two different growth fields, and so you cannot select it individually. And so that's a good question, right? But I mean, there's lots of ambiguity. So let's come to the sort of, what are the problems with this approach? The problems of identification. I mean, one very basic problem is, and I'm really surprised that a lot of geneticists never thought about this. Developmental geneticists, I interact most with developmental geneticists, but so if you tell people that perturbation-based functional approaches like genetics only yield necessary and never sufficient explanations, so you can only say these genes are involved in specifying the function, but you can never say these genes that I have are sufficient to bring about this specific function. Okay, so we are limited in this sense. But a really central problem here, and my philosopher collaborator, James DeFrisco, and I have just written a big paper about this, is the problem of correspondence, okay? So, for example, identifying the function or a trait is extremely difficult. So actually, the distinction of character identity and character state, for example, is difficult and heavily context dependent. So, think about cell types. There's a huge disagreement about how many cell types there are in a human body in the literature. We cannot sort of agree on what makes a cell type different from another cell type. And that's a very clear-cut sort of phenotype that you can look at, I think. So that's one problem. Individual characters often contribute to both identity and state, and more generally, of course, there's pleiotropy. I mean, a lot of genes factors contribute to lots of different functions. And homologous traits often are generated by different network mechanisms because of developmental system drift. So basically, there is no reason to assume that the genetic mechanisms that we find by doing this sort of functional decomposition correspond in any sort of rigorous way to the morphological characters that they are supposed to produce. Does that make sense? So that's a very important problem, very important limitation. And then I keep on going on. If you've seen a bunch of talks, I mean, this always comes up. The problem of diachronicity. So basically, static network diagrams never explain dynamics. Okay, so in the end, you end up with a collection of genes and potential interactions, but you don't have a temporal explanation of the phenomenon. Okay, so if you have a static graph of a network, that itself is not an explanation for the process, causal explanation for the process that generates the trait. It never is, period. Okay, so that's a big problem. You have to sort of make sure that you get from the necessary list of ingredients to a sufficient explanation of the observed dynamics that you actually see. So genes and any other networks based on genetic and molecular decomposition only provide a very rough first sketch. It's a necessary step. It's very important we identify that the parts that are involved in some sort of function we're interested in, of a true decomposition of the genotype-phenotype map in terms of functional modularity. Okay? It's not, I'm not criticizing it, saying it's wrong. I'm just saying it's not sufficient to give the kind of explanation that we want. And so people complement the functional approach, the genetics perturbation approach, with more structural approaches. Okay, usually. And most famously at the moment, if you are anywhere near the field of vivo, you've seen tons of drawings like this. They're even all made with the same software nowadays. Eric Davidson, and this is Eric Davidson's famous endomesoderm specification network in sea urchins. So the idea is that we look more systemically at the structure of a network, and we infer modularity from the type of wiring and the position of different connected components in the network. Okay, that's quite simple, and it should be quite familiar to everyone. Okay, for example, in Davidson's framework, you have these genes. You see there's not quite as many genes as you think here because they're repeated in different places, but these very conserved genes and their interactions, they constitute the kernel of the endomesoderm network. A kernel is simply the most highly conserved part of the network. This is another ubiquitous characteristic of evolving complex systems. Bill Wimsatt has come up with the term generative entrenchment. That means that some parts of the network are just going to be entrenched after a while. They become so important and central to the network that they can no longer change. And so these sort of central parts of the network that are very conserved are called kernels in Davidson's framework. He likes computer languages, so there are parts of the network with signaling components like the notch pathway here that mediate between kernels and other parts of the network, and they're called switches or input-output devices. And the computer metaphors go on. Further down, you have these sort of plugins there that are locking a tissue into a specific state, like into the skeleton fate, that is then executed by a bunch of differentiation genes. This is the skeletogenic differentiation gene battery. These transcription factors are involved in producing the actual skeleton structure, physical structure. And the idea here, of course, is that mutations in different parts of the network reflect different phylogenetic levels. So if you want to learn about speciation, you have to look at changes in these differentiation genes. And if you want to look at difference between phyla, you have to look at the kernels and how they differ between the phyla. And the explanation for the stable presence of different phyla is that after an initial burst of evolution, they became entrenched and couldn't evolve any further. Okay, so that's the sort of sense in which this sort of approach is supposed to give us evolutionary explanations. But it relies completely on the identification of structural network subcircuits. Okay. Now, how do we know what these circuits do? This is a different sort of decomposition of the network that goes much further into detail. It's from a paper from 2017, a posthumous paper by Isabel Peter and Eric Davidson. And so they dissect the network, these small components, and they assign functions to those. They say, okay, if you can dissect it to small enough subnetworks, you can assign a function to each one of those networks. Okay? So we have toggle switches, feedback loops, feed-forward motives, and so on and so forth. So the idea is that you can just, with the Lego bricks again, if you can dissect the network in enough detail, you can figure out what each part does, and then the function of the whole network is just basically an assembly of all these little subnetworks, if you do it the right way. Okay? Another way of identifying such subnetworks is Yuri Alon's idea of network motives. So you have very simple subnetworks. This is a feed-forward motive. People call this a feed-forward loop. It's not a loop. Okay? See the arrows? There is no loop. It's not a loop. A loop is feedback. There is no such thing as a feed-forward loop. It's a contradiction in terms. I get very upset when people use it. So I've become too much of a philosopher. Okay, so this motive has an input gene. It activates directly an output gene, but there's also a sort of indirect pathway that activates it. And the activation of the output needs both of these pathways, okay? And that's a persistence detector. Basically, if you have a little pulse of signal, this slow pathway will not react, and you don't get a reaction. You see, you get a little bump of Y, but never any Z. But if you have a persistent input, then a reaction is triggered, and so on and so forth. Okay, so you can come up with a whole zoo, and Uriel did that, of different types of these motives, and you can also do the statistics of different networks and say they have different profiles of you can count how many of these, how many of these, how many of these, and can say I can distinguish, for example, structures in the internet from regulatory networks in biology over here, because each one of them have what they call significance profiles, so distinct distributions of those motifs that are deviating from the null model that you would expect by a random sort of wiring of the network. Alternatively, you can go and just look at the structure. So it's called community structure. You have algorithms that detect regions of higher density in large networks. Here's an example of a protein-protein network. And if you have a more sophisticated approach, you can have overlapping sort of modules. And the idea is that if you find such sort of communities within the network, they will be more likely to do different things. Okay, there is a very simple problem with this approach. And this is that structure does not determine function. I have, in the paper that we wrote, we have a whole list of citations from Eric Davidson claiming that structure determines function. And you repeat it, and you repeat it, and you repeat it. No matter how many times you repeat it, it's simply not true. Even for a very simple network. So if you take this classical sort of 3-gene network, you have 3 genes represented by dots, and they're constitutively activated, and they repress each other. It's called a repressilator. Classical work in synthetic biology by Elowitz and Leibler in 2000. What does this little circuit do? Okay, so what you have to remember that in most cases, for most parameter values, it does nothing. Okay, so the default state for most of these networks is extinction. All of the concentrations just go to 0 and nothing happens. Okay, nothing interesting happens. So most network structures do nothing in the first place. But if you get lucky and you hit the right parameter, the right sort of space of parameters, this network can do 2 different things. It can oscillate stably. This is a beautiful movie of a mouse pre-Semitic mesoderm, and you can see waves of gene expression going through. So you can this is a negative feedback loop that can create stable oscillations. More often, the oscillations will go on for a while and then die out. But these are 2 qualitatively different behaviors that you can get from one single circuit. So you get 3 different dynamical regimes basically from this circuit. You get the nothing regime, the damped oscillations, and the stable oscillations. Now the problem is, if you've managed to identify the presence of such circuits in a bigger network. If you add just one interaction to this, everything changes completely. So let's do that. If we add one interaction, a back reaction up here, the circuit has a different name now. It's called the AC-DC circuit, and because it has a positive and a negative feedback loop. Okay, so it can oscillate and it can switch like an electric AC-DC circuit. We didn't name this, by the way. It's really cool. But I mean, apart from these 2 types of oscillations, once again, the default state is it doesn't do anything. But if you have the right kind of parameter values, it can, just like the repressilator, it can oscillate, or it can switch, or it can have what are called relaxation oscillations, which is basically a switch that goes back and forth. So by just adding one additional interaction, you've doubled the number of different behaviors that the circuit can implement. And you can repeat this for any sort of simple motive like this. Okay, even the simplest networks can do a lot of different things. Networks are multitaskers. Okay, so this idea of being able to look at the structure of a big network and dissecting it in some way and understanding what it does or what the subcircuits do and then putting it together again just doesn't work. Okay. And it's a bit frustrating to see the entire field moving into sort of showing you these big networks. It's an important first step, but then you need to find out what these networks do, and that's much more difficult than drawing them in the first place.
But you have to agree that, of course, I mean, it would make no sense to try to find the structure if some structures would not be better at doing some things than others, right? So there is an importance to the structure, just to not push it too far.
So the network structure constrains the function, but it does not determine the function. So you can narrow it down, but the problem is the context dependence is really quite radical. So for most of Fourier-Lens motifs, if you put them into their native context, they don't do anything like what they're supposed to do. So in the gap chains that I'll introduce in a minute, we can find about 13 different feed-forward motifs, and none of them is a persistence detector like they should be in theory. So that's the biggest problem, I think. The radical context dependent of the behavior of these circuits. So where do you draw the line around the subcircuit as well? Because the community structure is in theory a good idea, but it's very rare that we find very cleanly identifiable clusters of interactions in network graphs. So there's a problem of composition. Network behavior can be extremely sensitive to temporal or network context. The addition or removal of a single interaction can completely change the dynamic repertoire. We cannot expect the behavior of a complex network to be a straightforward composite of the behaviors of its modular components. Basically, it's not Lego. Evolution doesn't work like that. Okay. Also, by the way, a huge problem for synthetic biology is having a massive amount of problems. They created this whole BioBricks collection of circuits that you're supposed to just combine. It never works. It's exactly because of this. You put the network in a very complicated, self-regulating background. And you know, the danger of flogging a dead horse, it's still a static explanation. Okay, it's just a network graph. These sort of static explanations cannot explain developmental dynamics. So basically what happened is that by introducing this sort of network thinking, we've not only failed to resolve most of the problems that we have with genetics, but we've created a few new ones in addition, okay? Which is okay, I think so. So don't get me wrong, I mean, people always sort of ask me, then you're against sort of identifying networks. No, you need to do this. I'm just saying this is just the first step. And then you need to find out what the network actually does. But of course, you need to have the parts before you can understand the behavior. But there is only a loose association between network structure and function, and that's a really big problem. So we cannot just focus on structural modules. Just to illustrate this a bit more, there's a beautiful study. This is a modeling screen. So these people took an ensemble approach where they model. So they come up with a two-cell model that has a signaling interaction between the two cells and then a feedback interaction between the two genes. It's kind of a simple model of maybe a notch delta pathway that can distinguish, you know, switch this cell on and this cell off, sort of an on-off switch. And then they play around with the strengths of these interactions and they simulate how do these models behave. But the simulation has a twist. They simulate it in two different tissues. First in a tissue that has no contextual signal, and the second simulation is in a tissue that gets a signal that changes the behavior of the network. Okay? And they want to find out. So in natural systems, the notch delta pathway has two very different behaviors. One is called lateral inhibition. So whenever a cell switches on the pathway, it'll inhibit all its neighbors from expressing the pathway. So you get this salt and pepper sort of pattern of regularly spaced cells that are switching the pathway on and cells that have the pathway on. That's called lateral inhibition. But it can also counterintuitively do this. You have a line of cells and they switch their neighbors on, okay? So that's exactly the opposite of this behavior called lateral induction. And the question here was, can a simple circuit like this exhibit both of these functions just because in one case there is a signal present and in the other case there isn't. So when they did this screen, they found lots of different circuits that could do this, okay? And they analyzed all of these circuits. And they found a really interesting, so they analyzed which parts of the circuit are contributing to which function. Okay. So here is a rather complicated graph. You have a full network here, for example, you can think about this. And the two dashed lines here indicate the components of the network that contribute to different functions. You could have, in the best case, this sort of situation where you have two parts of the network and they contribute to the different function. These are called hybrid circuits. Okay, so this is where structural decomposition works. But they found out most of the circuits were not like that. So they had some sort of overlap between those modules. Or, in the most embarrassing cases, they had every component of the network involved in both functions, but behaving in a completely different way in different contexts. These are called emergent circuits, and there's sort of a big spectrum of multifunctional circuits that they found, but most of them were falling in these sort of categories. So this simulation study seems to indicate that if we only look for structural modularity, we will miss most of the circuits that can be multifunctional but have no structural modularity or very limited structural modularity. So it's not wrong to look for structural modularity. We'll just miss a lot of the cases that would be interesting in terms of, so it means that circuits that have no structural modularity can still be functionally modular. Does that make sense? So one of the reviews of the paper that you read this week apparently was saying that this is their modularity is no modularity. Okay, so I want to be very precise here. So I'm saying, yeah, modularity is more than just structural modularity. So in terms of its behavior, this network is still modular. It does two different things which you can distinguish from each other and which you can also, I will argue, select.
Is that question to clarify?
Yeah.
So when it does two different things, so that's with the same parameter values?
No, so each one of these solutions has a different set of parameter values.
Right.
But they do with the same parameter values. The only difference is the presence of a signal or the absence of a signal. So yeah. The only thing that changes are the boundary conditions.
So it does the two behaviors depending on the
same parameter values.
So the same parameter values for the activity.
Yeah, that's how they fit. So it's the same network that does both functions, but the difference is only the signal coming in or not. So most circuits are structurally not stable, but still multifunctional. Okay. So this is just virtual. So let's look at a real sort of circuit. Before we do that, I would like to introduce this idea of dynamical modules. Okay, it was first introduced in a paper by David Irons and Nick Monk in 2007. They say rather than being a protein complex or a group of co-expressed genes, such a module can be viewed as the temporal activity of a group of genes or proteins that controls a specific function in different environmental conditions, cell types, and/or tissues. Slightly simplified, dynamical modules consist of a set of network nodes connected by regulatory interactions that implement a particular type of behavior exhibited by the larger network they are embedded in. So basically, they are a component of a larger network that does something specific. That's basically it. And they don't have to be modular, and they can overlap. Okay, they don't have to be separate. Okay. So Irons and Monk used a very simple example with a Boolean network model of 6 genes. So you see here, these are on-off. Each one of those circles is an on-off switch, and the arrows show you whether, so if number 1 is on, it'll switch number 2 on, number 5 on, 5 will switch number 6 on, number 6, if it's on, will switch number 5 off. Okay? It's a small toy model, and you can completely enumerate all the possible behaviors from all the possible initial conditions of this model. So you have rules, you can formalize this, and then you can find all the different attractors of the system. So the system will settle into specific patterns. And what you see here, this is the simplest pattern. All genes are on and stay on. And this pattern will switch over time through these different stages and then start again. So these are cyclical attractors. These 4 attractors are all the attractors of the system. And now you can sort of dissect these these different behaviors. So basically, you take the common elements of these attractors and say, okay, so this is like Lego now. So you can take these attractors and say they are based on stereotypical behaviors, and all of those attractors are a combination of these different subsystems. And you can map these subsystems back onto the network. So, for example, subsystem number 5 is this module is implementing a certain behavior, and the overall behavior of the network is just a composite of those different behaviors.
Just clarification. So
Yeah.
the C0, C1, that's times.
So this is time,
Yeah.
and then x here too is time, and the numbers are the different genes.
Yeah.
So that's just to give you a basic idea. So you map. First of all, this is an algorithm, so you can run it on any Boolean network, and it will give you the subsystems, this set of subsystems that creates the set of attractors that you have here. You can run it on more.
So what do you mean by A1, the attractor? That is a cyclic attractor that
Yes.
goes through those 4 stages and back
Exactly.
again. Huh?
It cycles through these and then again here.
Yeah, okay.
And it's basically a composite of this subsystem, and these two subsystems here. So these are the greatest common denominators of the different attractors. Does that make sense? And you can run it in a Boolean model. You can run it through an algorithm. You can take a Boolean model of the segment polarity genes, for example. This is a Drosophila embryo. This is the head. This is the tail. And you have these cells, and at each boundary of the segment, you have a sort of an on-off switch between one state expressing wingless signaling molecule and one state expressing the engrailed transcription factor here. So if you have a boundary between 2 segments, you always have this on-off pattern. And you can make, I'm going to run very quickly through this just to show you, this is a very complicated model. You can identify the attractors and the subsystems, and that gives you certain symmetries of the whole system. So basically, you can immediately understand and simplify the behavior of the entire system by looking at these different subsystems that implement different behaviors. Okay, I don't want you to stare at this for too long. It's just to show you that if you have a Boolean model, there's an algorithm and you run it on that, and you can identify these subsystems that correspond to the dynamical modules of the network. Now, if you have a more complicated model, you have a morphogen gradient-based patterning like in the earlier Drosophila embryo, you have a problem, okay? Because you cannot just generalize this for such more complicated systems. So what we've done is an empirical investigation into a particular patterning, dynamical patterning module, which is the gap gene system in different flies. I've briefly introduced the segmentation genes before, but just to tell you again, so there's a bunch of maternal gradients. This is the anterior of the embryo, the posterior of the embryo, and these gradients instruct a bunch of overlapping gap genes that regulate each other, if you remember. And you can look at different species. This is Drosophila melanogaster. The red flies here in the phylogenetic trees are the Cyclorapha, unfortunately named higher flies. I think mosquitoes would object to that. And then the second big branch of the Diptera are the mosquitoes, and you have midges down here. And you can see that all of these have very similar gap gene patterns, but have quite some variability in the input in the maternal gradients. So we were interested in comparing these two different species and trying to find out if we could understand the compensatory evolution that happened here, and also the different dynamic behavior of the two systems by dissecting the system into these dynamical modules. Does that make sense? So here you have to stop me. This was the paper that I guess we'll go over the paper that you
No, we didn't take this one.
the other one. I'll talk about the eLife paper.
Yeah, okay.
Just before I do that, the method is a little unusual that we use. So we have a reverse engineering approach where we quantify gene expression patterns in different species. So we measure them, we quantify them, and then we come up with a mathematical model. This is the only equation I will show. This is derived from a neural network model, a very, 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 very simple gene network model. Okay, and it's a differential equation model that basically describes how the concentration of each gap gene changes in each nucleus, and you have these dividing nuclei in the embryo here over time. And the concentration changes in reaction to protein decay, protein diffusion, and then this big spaghetti of an equation here, which is basically just a sigmoid switch function here, the theta, and then a large sum of inputs. So all the inputs from the other regulators are fed into this function and then result in a certain expression state. So you have, in the model, you have a matrix of interactions. So each number here corresponds to a specific genetic interaction. This, for example, is the interaction of regulator number 2 on target gene number 1, and so on and so forth. So it's a really abstract model that doesn't consider any of the molecular details, and is formulated in a way that is simplified. There's only one threshold for each gene, and the aim of this model formalism is that we can fit it to the data. Okay, so this is neural network models are basically built to learn, so we can learn the network from the data. And the way we do it is we plug in a random network, we compare it to the data, it will give us something that doesn't look at all like the data, and then we mutate it, and then we select for better fits, and you do that a couple of million times. You're literally evolving the model in the computer until it produces exactly the patterns you see here, and then you go back and you look at the interactions. That gives you a structure of the network and also dynamical behavior represented by these phase-space diagrams. We won't need so much of those. So basically, you're learning the network from the data. The very depressing thing here is, you know what the most efficient evolutionary algorithm is for this really complicated problem? It's called scatter search, and it's a sort of a intelligent design. It basically explores the parameter space in very sort of directed ways, and then uses selection based on that. So intelligent design works for model fitting. It's much better than random evolution. Okay, so that was totally beside the point. What I wanted to say is that we get a model that reproduces the data very, very closely, and we get a network, and I'm displaying it in a slightly unusual way. So this is a spatial representation of the embryo. You have the anterior to the left, posterior to the right, and these boxes represent the different expression domains of the gap genes. Okay, that's why they're repeated. There's an anterior hunchback domain, a posterior hunchback domain. And you can see that there's two pairs of non-overlapping gap genes, and they have very strong double negative, that's a positive feedback loops. So these are switches, okay? So they either this one is on or this one is on, but never both. And then you have a sort of a bit of repression from the posterior. The background color represents the activation from the maternal gradients, and you have this very mysterious layer of much weaker interactions between the gap genes, and nobody ever knew what these were about. The dynamical model tells you that they are regulating the dynamics of gene expression, which is you have genes that are shifting. I'll show you in a minute. And we did this in both flies, and you can see that we find basically the same network in both flies, but the model allows us also to look at the strength of the interactions. So there are very subtle differences between the two species. This was at first a little disappointing. I mean, this postdoc invested 6 years of his life making this model for Megasilia, and then we found the same network. Okay, that was a sort of a day of heavy drinking after lab work. And but then actually we thought this is really interesting because we called this quantitative developmental system drift. And the idea is that you will always in evolution have a sort of a wiggle room in terms of the strength. So there's a lot of different transcription factor binding sites, and they have quite fast turnover rates in evolution. So you will get a constant fluctuation in the strength of these different interactions, even in the same population. Okay, so there's a lot of sort of room for compensatory evolution there, I think. Okay, and so that's actually very interesting. And even though these differences are very small, that doesn't mean the effects are not important. And actually, we had a reviewer that wrote, the differences are small, therefore they are not important. Which, you know, that's just... okay, because these small differences between the species explain very big differences in expression dynamics. What I haven't told you yet is that, so there's two different modes of gene expression. In the interior of the embryo, here you see 3 expression graphs, the model in dots and the data in filled areas, and the time procession over time. So you can see here in the anterior of the embryo, the domains, they stay the same, the boundaries stay in the same place, while here in the posterior, they shift systematically towards the interior and all the domains become narrower and narrower. Although both of them look very similar, they're based in 2 completely different mechanisms of gene expression. This one is a switch-like system where different nuclei fall into different attractor states, just like in that logical model I was showing you before. While in the back here, to everyone's surprise, you have a clock. And this is very confusing. There is no oscillating gene expression here, okay? There's just a very bad clock. You have to think about these waves as like Mexican waves in a football stadium, okay? Nothing moves. People raise their hands, so the wave moves through the stadium, but none of the spectators have moved. The same thing happens here. So each nucleus switches through the expression of different genes over time. And if you put that in a spatial context, you get the impression of movement, but nothing actually moves, okay? So it's just nuclei switching on and off different genes in a stereotypical way. So you have crippled and knurped and giant and hunchback. And if you would give them enough time, they would all end up expressing hunchback. But all of them start this cycle at a different time. And so if you put them together, they all express a different sequence of genes at different time, and you get this apparent kinematic motion of the, okay? So very different behaviors here. Just for those who are, here's a metaphorical description of the behavior, just to show you how different it is. So here, the nuclei are pretty set from the beginning. They fall, wherever you start, you end up in a specific end state, while this is a system that's much less deterministic. If you have children yourself, you know that this is a classic example of a damn oscillator. And you have to imagine this is just a clock that never completes the hour, or a child that just swings once and then stops. Okay, but qualitatively, the take-home message here is these are qualitatively very different ways of dynamical behavior. So we wanted then to map these different behaviors back onto the network. And we were stuck in this paradigm of structural modules. We looked at the network and there's no structural modularity. You can't find any community structure. And we tried to map the behaviors back onto the network until we realized that the answer was in our face the whole time. So basically, if you look at this network diagram, in the anterior, the middle, and the posterior of the embryo, there's only ever 3 genes expressed at the same time out of the 4. And if you look closely at how these different sets of genes are connected, you notice that they're always connected in the same way. So all of these circuits, they're heavily overlapping. Every neighboring circuit shares 2 out of the 3 components with the previous one. That's one thing. And all of them share this sort of network topology, this network structure, which is, of course, the AC/DC circuit. And remember that I said the AC/DC circuit is the simplest circuit that can make switches and oscillations. So this is what we have in the system. So you can map these circuits back. And you can look in the models for the different species, what are they actually doing? So in Drosophila, the anterior-most circuit is in a switch regime. Okay, so it is in the behavior that produces stable switches. I'll come to that in a second. And the one in the posterior oscillates. This one, we were thinking, okay, different circuits will correspond to different behaviors, but this one is critical, meaning that it does both behaviors. So it gets different inputs from the maternal gradients, and it can either switch or oscillate. And it sits right on the boundary of those two behaviors. Okay? So now think about this in terms of evolution. This one is the most sensitive. We were talking about sensitivity analysis before. So these two are robust, and this one is sensitive. Okay, so if you change any of the parameter values in the system, this circuit is going to get hit or has the most effect. Okay, if you change the parameters here or here, it doesn't matter so much. And the funny thing is that you have interactions in common. So if you change this interaction, it will only change the behavior of this circuit, but not of this circuit. It's very counterintuitive. So what happened in evolution is that in Megacelia, this circuit became critical, while these produce oscillations. And this means that in Drosophila, the boundary between the two different behaviors is in about the middle of the embryo, while in Megacelia, the boundary has shifted. Okay, so you have a lot more oscillating patterns. And you can show, we have some models in other flies, that the position of this boundary and the dynamics of these domain shifts is very labile, while the sort of the general setup of the different domains is very hard to perturb. Okay, so basically you have a very conserved spatial temporal arrangement of gap domains. You can hit the models really hard and nothing changes. But this sort of boundary, the bifurcation boundary it's called, between these dynamical regimes is evolutionary very labile, and therefore you have differential evolvability, and we'll have a discussion about what I mean with this here, of different expression features. It means that you get random, you put random variation in and you get non-random variation out of the system, and you can now map exactly what sort of features of expression are most likely affected, and you can then also, actually this is work I'm not showing you here, but you can show that this regulation of these shifts is very important for the compensation of variability in the maternal gradients that are coming into the system. This is why this system is perfectly suited for filtering out variability from upstream inputs.
So what do you mean by differential evolvability? In this case, is differential between the different regions?
Features.
Between the species, yeah?
Features, not the species, yeah. So different features of gene expression have different sensitivity to parameters. So here evolvability means nothing but, it means two things actually. One thing is a non-isotropic distribution of the effects. So some parts of the network are much more sensitive than others. But it also means that you can dissociate the effects. So the interesting thing is here, you can mutate specific interactions, mutate in the model, but depending on which of the circuits is critical, it will only affect a specific circuit. So although the model, and this is a crucial point, but it's really hard to understand, although the model is not structurally modular, it is dynamically modular. Okay, so specific changes in the interactions will have very specific effects that only affect those modules that are in a critical state, unless you push the system really far and then everything breaks down. So my argument here is that you don't need structural modularity to have functional modularity. And this is a case study of that.
I totally agree. I don't think that it's actually required in many concepts that are dealing with modularity.
Absolutely.
Variational modularity is not, doesn't require the structural modularity.
Variational modularity doesn't require it. So for quantitative genetics, this is not a problem because you're not worried about the details. But functional modularity,
right.
No problem.
But that's a little bit of a thing here, because you're now treating these three sections as three functions.
Mm-hmm.
If you're applying this in the model, and I think that's a little bit of a little bit far-fetched, right?
It's a bit of, I wouldn't call it far-fetched, obviously. I would say it's a complication. And so if you do it in the Boolean model, it's very clean, black and white, because you have on-off, you have an algorithm that identifies different parts of the network that does different things. Here, where you have continuous concentrations, why would you expect that sort of thing, right? I mean, so it's going to be, it's very fuzzy, the whole thing. So basically the overlaps are massive between the different modules. Also their areas, I should say, the regions of influence where these are active obviously have massive overlaps because we always share two different expression patterns here. So basically there is a fuzzy modularity here, near decomposability.
Yeah, in terms of effects, but what I mean in terms of phenotype, you know, if we are saying if you don't need the structural modularity to have a functional modularity. And what I'm thinking is that the functions here are implied by the where the genes are expressed, right?
Kind of, but no. This boundary lies right in the middle here. Okay, so it's not. There's there's not a one-to-one correspondence. It's not at all obvious. Okay, so this boundary happens somewhere here in the middle, in this fly, and then somewhere much further here in this fly, and there is no one-to-one correspondence. It's not just simply the expression domains. Okay, it's more complicated than that. So if you have a continuous model, the bifurcation boundary doesn't have to. That's the thing about Boolean models. It's either on or off. So the boundary has to be between the on and the off state. But here it's much more.
No bad.
So I'll just put it in the share.
You have to structurally connect it to the German function.
Well, okay. I missed that.
What did you say?
You have to structurally connect it and then
Oh yeah.
determine the required function.
Can I do different things?
Okay, I'm almost done actually. I'm probably way over time already. Yeah, so a wrap up for the dynamical models here. Dynamical decomposition so this is important, is a top-down approach, partitioning the network into interlocking subsystems. It's not like the Alon's motifs that go from bottom up. Okay, so we are looking at the behavior of subsystems in their native context. And the behavior of the full system does correspond to a straightforward combination of the behavior of its models. But not straightforward in the way that we expect it. Okay, not like the Lego bricks correspond, you know, little neat modules in a structure correspond to different behaviors. I just said that. Dynamical decomposition allows us to study the dynamical behavior of subcircuits in their native context. So dynamical modules are a much better proxy for functional modularity than structural modules, since dynamics is much more closely related to function and structure. So basically, most of the theories on pleiotropy, for example, depend on functional modularity, not structural modularity. Okay, and this is a big confusion in the field, because I think in Evo Vivo especially, I don't know outside, it is often assumed that you need structural modularity for evolvability. You don't. That's the point. Take-home message here. So just a little advertisement that they're not just in gap genes. Dynamical modules are present in the vertebrate neural tube, which is, to my delight, full of AC/DC circuits. That's where the name came from. Turing pattern generations are dynamical modules. And there's a beautiful piece of work from James Sharp, a paper you should definitely do for Journal Club at some point. Ko and Maru, a postdoc in James' lab, dissected dogfish embryos, made little cuts in these mermaids' purses, injected them with ink, waited for 2 months, made a fate map of those embryos. Beautiful work. A man of endless patience. Absolutely beautiful work. And so they made a model of the limb growth in dogfish that English people confusingly call cat sharks. And mice, and they were showing that the different patterns of the distal elements in these two species that were thought not to be homologous. So this is the sort of digits of tetrapods are one of our most proud examples of an evolutionary novelty, are not a novelty at all. they are just too derived to recognize them. They could show that the pattern generator is very conserved, in fact, but produces completely different outputs, and they were able to homologize these distal structures. This is very controversial, especially around the KLI, for example. But I think it's very.
Yeah, but one would question whether gene expression has been questioned as a evidence of homology, and
so this
networks are just one.
This is like a pre-pattern, right? That prefigures the actual chondral condensations. But I think it's pretty cool. It's created a lot of discussion. I'm sitting on the fence. I don't have any investment in invertebrate limb evolution, but I think it's a pretty nice way of showing that you can use dynamical modules not only to explain development, but you can also use them for sort of these lineage explanations, plausible scenarios, how you get from one structure to the other, basically. But these, of course, these dynamical modules, they're nothing else but morphogenetic fields, conceptual foundation of classical embryology. Okay, it's the same thing. They also exist in plants, but more importantly, they go further than morphogenetic fields. You can consider the cell cycle network as a module, dynamical module, or circadian rhythms. So they're really everywhere in biology. And also here, there's no obvious modular structure to the networks, but you can dissect them in terms of their function. Okay, to wrap up, I'm way over time probably. Functional modularity of the genotype-phenotype map is a prerequisite for the evolvability of character traits. Nothing changes. This is just a fact. Most regulatory networks are multifunctional. They implement more than just one dynamical behavior, even very, very simple regulatory networks. Most multifunctional networks are not structurally modular. So it's okay if you find structural modularity, good for you, but you're going to miss most of the functionally modular networks. Extensive structural overlap between dynamical modules implies that many genes and their interactions contribute to more than one dynamical behavior of multifunctional networks. Even though genes and interactions are shared between modules, their effects on the dynamical behavior of each modules are different. So it still fulfills this condition for it to be a basis for limited pleiotropy. Okay? So you can have limited pleiotropy even if you don't have a structurally modular network. So we have an alternative explanation for limited pleiotropy, and the structure of networks and regulatory sequences does not necessarily have to be modular to ensure dissociability of functional module. The prevalence, and this is very important, the prevalence of modular structures in the literature may therefore result from ascertainment bias rather than a true reflection of the organization of regulatory systems. We find modules where we look for them in network structure. Also, cis-regulatory elements are not modular. But we think they are because those are the only cis-regulatory elements we find, okay? But there's no evidence that cis-regulatory elements are truly modular. There's actually more and more evidence that sequences outside the series play very important roles in regulatory evolution. So, the identification of subsystems responsible for conferring character identity, to come back to that, can provide a powerful conceptual foundation for a mechanistic theory of homology. I was delighted that Günther is thinking about CHIMs now, character identity mechanisms. I hope he's going to talk about that next year at the Evo meeting. Both functional genetic decomposition and approaches based on structural modularity fall short of reliably identifying and delineating such subsystems. That's very important. Dynamical modules are better. The dynamic behavior of a subnetwork is much more closely related to its function. So that's the reason why they are better. It's a lot of work to find out what these modules are, but it's worth it. Dynamical modules, morphogenetic fields, provide a causal mechanistic explanation of the generative processes underlying a character trait, accounting for its variational properties. So this is also underlying variational modularity. It's just a different aspect of the same phenomenon. And therefore, morphogenetic fields, rather than chain networks or structural modules, provide the causal mechanistic foundation for trait homology. And luckily Günther isn't here today, so I can say that. So we were just talking about this absolutely wonderful and undercited paper from 1999 in the JSB, Bondasso and Monroe. They say, rather than view dissociability as a problem for comparative biologists, let us recognize it as an architectural feature of evolvable developmental systems, a feature whose origins and consequences deserve attention. So this is such a wonderful paper. It's so long, but it has so much stuff in it, and it has very few citations. I recommend that you cite it. And I just wanted to sort of acknowledge the tons and tons of people who are. So the problem with dynamical modules, this is a lot of work. I spent 20 years of my life working on gap genes, and I've had it. This is why I'm changing to philosophy. I've had it and lots and lots of people over these years were involved in sort of pioneering the methods we use for modeling, and also getting the evidence, encouraging work in other species and so on and so forth. And I want to acknowledge the institutes that have been recently supporting me and the people who I still collaborate with at this. Thank you very much.
Okay, so we are over time, so I think we should more or less close the session there, and then we can have informal discussion. But the people who need to leave are
I stopped the recording.
yeah. So I think we can do that.